Hello, and a very warm welcome to today's ISNTD Connect webinar. It's my pleasure to welcome you here today. I'm Marianne Comparet from the International Society for Neglected Tropical Diseases. I'm speaking to you from beautiful, sunny London. It's our third sunny day, so us Londoners need to talk about that. It doesn't happen often. It's my greatest pleasure today to welcome from another sunny destination, the Balearic Islands, Dr. Claudia Paredes Esquivel. Claudia, a very warm welcome to today's ISNTD Connect.
Hello, Marian. Very happy to be here.
Hi. And Claudia, you are tuning in from the Laboratory of Zoology in the Department of Biology at the University of the Balearic Islands. And we are very excited today to hear a bit more about your huge body of work in this very mysterious aspect of One Health. Today you're going to be talking to us about the rat lungworm and why this is a One Health puzzle. We'll be focusing over the next 60 minutes on the lesser-known zoonotic parasite, Angiostrongylus cantonensis, also known as the rat lungworm, which has been increasingly found in the Balearic Islands. And this does have quite a substantial interest in terms of public health because angiostrongyliosis is considered an emerging zoonosis and is the main etiological agent of eosinophilic meningitis in humans. So without any further ado, Claudia, I would love to hand over to you for your presentation. We look forward to finding out all about this puzzle, and to all our attendees from around the world, again, a very, very warm welcome. Karina Mondragon-Shem, Natalia Moore, David Rollinson, James Lacourse, Carlos Teixeira, Helena Hamby, and many, many more. Hello and welcome back. And please don't forget, as we go along through the presentation, to add your questions and any comments in the chat function. So we look forward to hearing from you, Claudia. And again, a very warm welcome to today's session.
Well, thank you, Marian, and for the introduction, and thank you for giving me the chance to talk about our research here in the Balearics. Thank you as well to people who is in the public who is attending this presentation. For those of you who don't know me, I'm originally from Peru. I studied biology at at San Marcos University in Lima. And later I traveled to Europe to do a PhD at the Liverpool School of Tropical Medicine. I have mainly worked with other parasites, to be honest. I work with Chagas disease, with malaria, and also with other vector-borne diseases. So I come from that world, from the world of vector-borne diseases. So my field of research has changed dramatically in the recent years because of the arrival of the rat lungworm. This parasite, Angiostrongylus cantonensis, is in many aspects is a terrifying organism, but it's also a very fascinating one. So I hope you enjoy the presentation. So let me tell you about our beginnings. So as Marian said, I work at the University of the Balearic Islands in beautiful Mallorca here in Europe. And Mallorca is an important touristic destination in the world, particularly in this region. And we receive millions of tourists each year. This is one photograph of our beautiful beaches. So we never expected to find a parasite that was so dangerous here in the island. So as you may imagine, because of the importance of the Balearic in the tourist industry, the arrival of the rat lungworm here has resulted not only in many scientific questions, has not only opened scientific questions, but it has also resulted in some social challenges. So I'm going to tell you more about this in a minute. So how did it all start? So in 2015, we started a close collaboration with the Mallorcan Wildlife Hospital, COFIB, and we wanted to explore the parasites that were infecting wildlife species, basically because there was not much done in the islands, in the archipelago. I mean, there were some studies in Ibiza, but they were like 20 years old. They had some years already. So we wanted to explore which parasites our mammals particularly had. And we were particularly interested in those parasites that were zoonotic because of the One Health concept. So now I'm going to show you what we found at the beginning of this voyage with the rat lungworm in these little mammals, hedgehogs. Let me warn you that the next video is going to contain is going to show the symptoms of these animals. So if you want to look away for a few minutes, for a minute, which is what it takes, please feel free. So in October 2018, 3 hedgehogs were admitted to the Mallorcan Wildlife Hospital with clear signs of a neurological disease. So this is what we observed at the time. So as you can see, the animals showed, in general, behavioral decay, involuntary movements, instability. Their muscles were atonic, meaning without strength. And in some cases, we observe also paralysis. Okay, so the video has finished. So this was the beginning of our journey with the rat lungworm here in the Balearics. And it was important for many reasons I'm going to explain you in the next slides, but also because this was not only the first detection in the island, in the archipelago, but also the first detection in this part of the world. In Europe, the parasite hasn't, the disease is not endemic. Europe is not endemic for the disease, for angiostrangulasis. And so this was one of the first detections in the continent. So now let me explain you more about the basics. Now let's talk about the parasite itself. So what is Angiostrongylus cantonensis? Angiostrongylus cantonensis is called the rat lungworm because it affects rats, which are the definitive host of the parasite. So the parasite is called a barber pole nematode because its appearance reminds us of a barber pole. You know, these symbols that barbers have in some parts of the world. So this is a beautiful photograph by Cowie. Cowie's group have been working very hard in Angiostrongylus cantonensis in Hawaii. And as you can observe, because of the disposition between the reproductive and digestive organs, the morphology reminds us to this barber pole. Okay, that is why it's called like that. And this parasite will have a complex life cycle that will include many different hosts. So I have decided to divide this cycle in one part to make it a bit more clear. So this is the life cycle of the parasite. And as you know, As I told you, rats are the definitive host of the parasite. So the adult parasites will live in the right ventricle and also in the pulmonary arteries of these vertebrates. Here will reproduce, and also they will lay eggs. These eggs will transform into larvae, and these larvae will pass in the feces, with the feces, into the environment. These first stage larvae will be in the environment, and they will be eaten by gastropods that are the intermediate hosts, meaning the parasite will continue the life cycle inside them. So the parasite will evolve into a third stage larvae inside these animals, these invertebrates, and then the life cycle continues when other rats, or the same rat, ingest these invertebrates and get infected again. Inside these newly infected rats, the parasite will have tropism by the brain, so you have a tendency to go to the brain, to the nervous system, and from here they will mature into a subadult and then travel to the lungs again, where they will continue the life cycle as it is. So far the life cycle is simple because it involves these vertebrates and these invertebrates only, but it gets more complicated than that. Because many vertebrates can eat snails and slugs. Even humans can get infected. So when we eat them, for example, when we eat contaminated gastropods, we also get infected, but there is an important difference, which is that the parasite is not going to be able to reach the lungs. It will stay in the central nervous system, causing eosinophilic meningitis, and because it moves, it's in a location that is not the normal location for the parasite, so it dies as well. And when it dies, it causes inflammation and a severe disease. No, so it doesn't only happen in humans, it can happen in many other vertebrates. But to make it even more complicated than that, there are some other animals, other invertebrates and vertebrates that can act as parathenic hosts. A parathenic host is an animal that will transport a parasite. But inside this host, the parasite will not evolve, will not change, but it will be maintained there. So it's a transport host. And in this way, it can also, if they are eaten by other vertebrates, they can infect them as well. Among them, we have, for example, freshwater crustaceans like crabs or prawns. We also have planarians, amphibians, reptiles, and the number is large. So for researchers like Lang, for example, this parasite is a promiscuous species. And I think it's a very appropriate name for the parasite, because the number of species that it can infect is very large. This is a review article by Spratt in 2015, where he, for example, record all these detections in different vertebrates. No, as you can see, there are domestic animals like dogs, horses, alpacas, sheep, but also wildlife, mammals like, for example, flying foxes, tamarins, and also marsupials. And even birds can get infected with the parasite. In the case of tawny frogmouths, these little vertebrates, nocturnal vertebrates from Australia, the situation can be even worse because some authors, like Ma, have discovered that in animals that are injured, there is a higher prevalence of the disease than in those that are healthy. which opened the following question, no? Is it a threat to wildlife? In the case of tawny frogmouths, it is, and it has been established like this. But what happened with all the other animal species that get infected, like hedgehogs? So to make it even more complicated, there have been some studies conducted several years ago, like for example, these from Wallace and Rosen. These authors conducted experimental infections that show that even fish were susceptible to the infection by the parasite. However, this was only conducted in the laboratory, and they haven't been found in nature. So we would need to conduct more studies in order to clarify the situation. In my opinion, it's much more unlikely because of the ecology of this species. But well, it needs to be studied. But it's also a promiscuous species because it infects a large proportion of the gastropod families that are present in many regions of the world. For example, this is a study by Kim and also by the COE group that has, this group has given us a lot of information on this parasite, a lot of valuable information on the parasite. They, for example, reported that 36 from 58 gastropod families that they can act as intermediate hosts. And they said, they maintain that almost any terrestrial or freshwater gastropods may have the potential to carry and transmit the disease. However, there are some particular gastropod species, like for example, Achatina fulica, the giant African snail, where the parasites seem to be particularly susceptible. Okay, it's because there is a large amount of parasites. They can harbor a large amount of parasites inside. So let me tell you now something about the disease. Human angiostrongylliasis, it is called, and it's also called neuroangiostrongylliasis. And as you know, it doesn't only affect humans, but also other animals. So, as I told you before, they will cause an inflammation which results in a meningitis in people. So in general, if it's symptomatic, it can be considered a debilitating disease, with patients describing their symptoms as incredibly painful. They can last for several weeks and stop, but they may also be capable they may also be present for several years. So they can turn into a chronic disease. It can turn into a chronic disease. And even when the mortality in adults is lower than this, the mortality in children can be very high, well, high in comparison to other parasitic infections, with 10% of children studied according to the literature, resulting in death. So there is a large list of clinical manifestations. No? As in many other parasitic diseases, we will have some asymptomatic patients and also people that will develop mild symptomatology. But in some cases, the parasite can infect them and cause severe clinical manifestations. So this is a general list that will include, for example, fever, headache, drowsiness, blurred vision, and other symptoms that are related to meningitis infection. For example, neck pain, stiffness, nausea, vomiting, and many other clinical manifestations depending on the region of the parasite in the brain. Now, weakness in the extremities have also been reported, paresthesia, meaning pins and needles, and excessive sensitivity, not only from the skin, but also for light, etc., have been also reported. This is a very beautiful figure from Mondragon Shem in the Backbitten blog, a recent article in Backbitten blog. So, but there are some cases where the parasitic the parasite can cause a severe disease, an even more severe. And in these cases, we can observe, for example, visual impairment, paralysis, coma, and death as a result of the infection. Okay, so in general, human angiostrongylliasis occurs after the ingestion of raw or undercooked snails. Of course, this is going to increase the risk of infection. So in areas of the world where snails are eaten raw, like for example in some parts of Asia, but also in some parts of South America and in some parts of Africa, the risk might be higher. So it's important also to say this because snails in many parts of the world are an important source of protein, and they are easy to obtain and cheap as well. So we cannot really stop people from eating these invertebrates. So there is a social problem that we would need to confront in case of the parasite arriving into these regions. However, not only cases occur in regions where snails are eaten raw. There are many other regions where cases have been reported in humans. For example, this is a case, well, there are many cases in Asia, of course, and many researchers have been reporting these cases in the literature. But there are some cases, for example, in the United States. This occurred in Houston, in Texas. This is a case in tourist, in a Swiss tourist. Many studies have been conducted in human angiostrongylliasis cases in Hawaii. In Australia, there are also several reports. And in many regions of the world where people do not eat snails, there are cases. So how are people getting infected? That's the question. In most cases, they will occur by accident, of course. Pieces of snails or small snails and slag, for example, contained in salads or in sandwiches, they can be ingested accidentally, causing infection. Unfortunately, in some regions of the world, people have gotten infected on a dare, like, for example, in Brazil, in 2007, in Australia in 2003, in Hawaii. This particularly happened when people decided to eat raw slugs. For some authors, there may be some risk as well in relation to slime, because if you analyze the slimes of infected snails and slugs, the parasite may be present. However, the risk may be much lower because the number is not enough to cause a severe infection. More recently, Modri's group have demonstrated that when the snails are drawn in water, the parasite will escape. The larvae will escape from these invertebrates and contaminate the water. So the question remains on whether this is also an important source of transmission or just an alternative, an unlikely source of transmission. So more studies need to be conducted as well. But the rat lungworm story and the story of its dispersal is a story of parasite hunters. Parasite hunters like Chen, who discovered the parasite for the first time in 1935 in the lungs of rats in Canton. That is why it is called Angiostrongylus cantonensis. It was called Pulmonema cantonensis at the time. And this apparently modest discovery turned out to be a very important one, because 1 decade later, the same parasite was discovered in the cerebrospinal fluid of some patients in Taiwan. So this was the beginning of the story, of the fascinating story of the dispersal of the rat lungworm in the world. But there is one story that fascinates me particularly, which is the story of Joseph E Alicata, this Italian-born scientist that was working in the United States as a rat lungworm hunter. He initiated a voyage in different regions of the world, and his voyage started in the Pacific Islands, in these beautiful islands, also very touristic islands. He traveled through the Cook Islands, Fiji, Tahiti, New Caledonia, Tonga. He traveled to all these places trying to find the parasite, and he found it. So thanks to his voyage is that we now know that the parasite was widely spread in all this region. He was not the only parasite hunter, but he was possibly one of the most important ones. So the disease was called Alicata disease because of him. This is one of his first drawings on the disease. It already showed how many animals were showing infection. But not only it included the Pacific Islands. Joseph Alicata traveled also he also looked for the parasite in Okinawa, Sri Lanka, Madagascar, Mauritius, India, Kenya, Mozambique, Tanzania, Ecuador, and Costa Rica. So in many of these places, he couldn't find the parasite. Like, for example, in Mozambique, and Tanzania, Ecuador, and Costa Rica, he couldn't find it. And apparently, it must have been frustrating at the time not to find the parasite. However, because of his reports, we now can know that the parasite arrived later on and it wasn't present at the time in these places. So this is the current map of Angiostrongylus cantonensis in the world. So the parasite has been traveling westward from these Pacific islands, also continental Asia and Australia. And it has been traveling it was detected in some islands from the Indian Ocean as well, also in some parts of Africa. And later, it arrived to the American continent. The first detection in America was reported in Cuba in the early 80s. And for some authors, this was the result of the political connections between Cuba and some Asian countries, like, for example, Laos, Cambodia, and Vietnam, after the Second World War. So traveling in boats, possibly rats, contaminated rats, brought the parasite to this Caribbean island. And from here, it started an invasion into the United States, first New Orleans, and more recently Florida and many other places. And from here also to other countries from Central America, many of them islands as well. And from here also to South America, more recently. Brazil in 2007, the first report, Ecuador some few years later, and more recently Colombia. Even more recently, the Canary Island in 2010, with Pilar Foronda as the parasite hunter, reported the presence of this species in Tenerife. And even more recently, in 2018, we found it here in Mallorca. So this map is not, it needs to be updated, of course. So we were wondering, because in Europe, for example, it has arrived through the islands of the continent, no? First the Canary Island and then Mallorca. And we were wondering if it's a coincidence or if this is a parasite of islands, because it seems to be in islands where the parasite spread very quickly. In islands, for example, in the Pacific Oceans, in some islands, the prevalence in rats can be up to 100%, like, for example, in Rarotonga and in the Philippines. It can be up to 100% in rats, which is very, very high. So we were wondering as well, what is happening in these endemic areas of the tropic? Because as I told you in the previous slide, many reports in humans come from tourists or, for example, in places where the parasite is present, but where people is not exposed to the parasite because they do not eat, they do not consume raw gastropods. So what's happening in these other tropical regions where people are more vulnerable because of their their eating preferences? So let me tell you a little bit about our own parasite hunting. As I told you, we found it in hedgehogs. These hedgehogs were showing some clinical manifestations that were compatible with a neurological disease. So we decided to explore what was inside. And I remember the call of my friend from the hospital, Jessica, and she said to me, Claudia, we have a parasite that you are going to love. And she was right. I mean, love maybe is not the name, but it's true. It was fascinating. I remember I asked her, so does he have a dark line inside? And she said, yes. And I was like, it cannot be Angiostrongylus cantonensis. First of all, it had never been reported in hedgehogs. And second, it was mainly from tropical regions, so it was unlikely that it arrived to Europe. So we look at the morphology of the parasite and it fit. It was Angiostrongylus cantonensis. But there was something that called our attention, which was that we observed the presence of eggs inside females. That was a bit confusing because as far as we know, because of the literature, reproduction only occurred in rats. So after some molecular analysis, we arrived to the conclusion that it was actually Angiostrongylus cantonensis, so we confirmed the presence of the parasite here in Mallorca. And there was another question, another scientific question that remained open. Can hedgehogs complete the life cycle? This is also important because hedgehogs are widespread in the continent. In Mallorca, we have a lot of them. So this would open new paths for the parasite in this newly endemic area. So we studied the cytochrome oxidase 1 to conclude that it was in fact Angiostrongylus cantonensis, and it was really interesting to see that our haplotype, the haplotype of our specimens, was 100% identical with those from Taiwan, Tenerife, Australia, and New Orleans. For some authors, this is the article we published, for some authors like Cervena in 2019, he reported that there was possibly an invasive haplotype that was a particular population of this parasite that has the capacity somehow of traveling and invading new regions of the world. So this is something that would require further exploration, and this is something that we are working actually with the Muldridge group. So what we never expected was to confront additional problems in relation to the arrival of the parasite. As I told you before, the Balearic Islands are an important touristic destination in Europe. And so we receive a lot of attention from the press, from the newspapers and from the radio and from even the television. And they were talking about this nasty parasite in the island. And in general, for example, the local media broadcasters were very responsible because they transfer the correct information into the public. But there were some that gave misleading information. Like, for example, this headline, let me translate it for you, like eating hedgehogs and snails that are infected, raw or not very well cooked, can cause meningitis. And in Spanish, this can be a problem because the name erizos means hedgehogs and also sea urchins at the same time. And people do eat sea urchins. People do not eat hedgehogs, but people do eat sea urchins. So it can cause some sort of panic in the public. Of course, showing a photograph of the Mallorcan delicacy escargots is not really a good idea. Also because they are very well cooked, so the risk of transmission is not really, it's unlikely. This is another one. In this case, it's in English. It says, who eats the following delicacy risks infections with brainworm, and it's misleading as well, because marine species have never been found positive to the infection. So it's also threatening people, frightening people with no need. And this is my favorite. Well, favorite for explaining it in a way. It's actually the opposite. It says for the first time, it is detected a worm that is capable of causing meningitis, and they show a photograph of a caterpillar. This is even more frightening for people that look at this photograph, right? So we are confronting some situations like these. I mean, they were even making videos in the German media, in some particular media, where they were showing a completely different story. So finally, I would like to tell you about what's the importance of the One Health approach in confronting the situation of the arrival of the rat lungworm into the Balearic and into many regions of the world. So in our case, One Health has been crucial, and I think it's important always to apply this concept, this approach, into the study of the rat lungworm. So as you may know already, the concept, the approach of One Health is to reduce the risk at the human, animal, and environment interface, because it maintains that the health of humans is connected with the health of animals and also with the health of the environment. That is why it's called One Health. So we were conducting some parasitological surveillance in wildlife, and this helped to detect this nasty parasite, this dangerous parasite for humans. So we immediately connected our information, we reported this information, and we continue doing so into the local hospitals, because we know that now the diagnosis and the treatment of this type of meningitis, the type of meningitis that the rat lungworm causes, it's different than the treatment that those meningitis caused by bacteria or by viruses require. So knowing that the parasite is present in the island was crucial to establish a strategy. But also it shows us that animals can be used as biosentinels. And also it shows us that wildlife recovery centers like COFIP or wildlife hospitals are very important to early detect these emerging zoonotic diseases. So in our case, we work with these hedgehogs, and we consider these are a perfect example of a good biosentinel. We continue using these animals to understand what's the current situation of this parasitic disease in the island. And we have been adapting some techniques, for example, tissue digestion techniques. And thanks to this adaptations to this technique, we have been able to more accurately determine the number of parasites per animal. Now, this is a photograph of my student Sophia, and you can observe here in this Eppendorf tube the presence of 64 worms from a single hedgehog, meaning that the parasite is well established and that there are highly infected gastropods. So we think that, in the case of hedgehogs, they are important sentinels. They are not the only one. It will depend on the region, in every particular region, to determine which biosentinels are the most appropriate to follow the dispersal of this parasite, and also to early detect the presence of this parasite. But it is important to work in close collaboration with these wildlife recovery centers in order to establish these methods, these strategies. Snail surveillance is also important. So establishing surveillance in invertebrates is also important, because some of them will carry also parasites like this, no? So we are also conducting a snail surveillance in the Balearics. We haven't analyzed the snails yet, because we are right now looking for some funding for this. But it's important, because in that way you can determine what are the most important species in a particular region that may be the main vectors of the disease. Like, for example, here in Mallorca and in Tenerife, it occurs the same. The giant African snail is not present. So we have other species that are responsible for the transmission in the island. This is a photograph from my country, from Peru. The giant African snail has arrived recently, and people is obsessed with eliminating them. However, they are not examining other snail species, like for example, the one I showed you before, these churros, which may be harboring the parasite as well. So studying the invertebrates, conducting snail surveillance, it's very important to establish to understand the real situation of this parasitic disease in many regions of the world. And the One Health concept also includes the food protection, the food industry, because, animals can also serve as food for humans. However, the situation is even more complex in the case of the rat lungworm, because this snail farm industry that we are having in many regions of the world, and particularly here in the Mediterranean, is growing. Okay, the number of farms is growing. There is more people interested in opening these farms because of the economical importance of this. You know that there are some cosmetic products that can be produced with the snails, with the snail slime in particular, and this is something very new and very popular between the most demanding customers. Snail caviar is growing, and the interest for obtaining snail caviar is also growing among snail farmers. So here, for example, in the Mediterranean, we consume snails, but they are very well cooked. So the risk of transmission is low or inexistent if they are very well cooked. However, there's people that is manipulating these animals, these invertebrates, in the farms. So we were wondering what kind of risk they may be confronting by doing these activities. So we are right now working in this small project funded by the Balearic agricultural section of the government, which is Caragolsa, which I invite you to visit our website, because here we are trying to collaborate with the snail farmers and also with the food protection agency in the island in order to establish appropriate control methods and protocols and establishing strategies in order to prevent the entrance of the rat lungworm into these industries. So far, we've been lucky to find no parasites in the snail farms, but we are trying to protect them even more. And finally, I think in general, it is well known that invasive species can damage the environment, can alter the ecosystems by competing with the native species, and they are in general try to be avoided in many regions of the world. But what we have to take into consideration as well is that invasive species may also carry silent invaders, like the rat lungworm, that can cause even more devastating effects. So campaigns like these are perfect. Of course, they require further investigation on the exact relevance of these species, but also by designing policies to protect ourselves from the entrance of these invasive species, we may be protecting ourselves from these silent invaders that they can carry. So finally, I would like to thank to you for attending this presentation. And also, I would like to thank to the Angio team and all the organizations that have been collaborating with us, like the local hospitals on Espaces and medical doctors that work there, LSTM, our university, particularly the Oficina de Soporte a la Recerca, Fogaiva, and also our group, Miguel Ángel, Carlos, Ana, and the rest of the people from the zoology group, but in particular to my students, Sofia, Victor, and to some ad honorem collaborators that we have that work also very hard in the study of this parasite. Sebas, Ricardo, who is also a firefighter, Neus, who is also working with us, analyzing other wildlife mammals. And I would like to thank particularly to Lee and Karina, who are from LSTM, and that they have given me important feedback for the construction of this presentation. So thank you very much, and please contact me if you have any questions.
Thank you very, very much, Claudia. That was a really exciting talk, and it was amazing to see kind of the whole scope and breadth of the parasitologist's investigation there, all the way from the literature review to the surveillance, also that kind of difficult relationship with media outlets that you described, and trying to kind of elucidate through all of that a situation that is very clearly evolving rapidly. And you're getting a lot of thank yous in the chats from our attendees as well. Majid Fasihi Harandi, thank you very much for your presentation. Eric Morgan, fantastic talk, thank you. James Lacourse, wonderful. Claudia, absolutely, and we most certainly echo that and feel that very strongly. Claudia, I don't know if you probably didn't have a chance to see, but during your talk, there was actually quite a few questions coming up in the chat, a bit of discussion, particularly around diagnostics and surveillance and so forth. But I suppose one of the things that I think a lot of us will be wondering is where do you think we might find the rat lungworm next? Are there other areas, for example, in Europe, where you feel the parasite may be present? And in fact, Eric Robinson was also asking, how do you think that this parasite arrived in Europe? I know you outlined a few of those hypotheses throughout your talk, but where next for rat lungworm? Where should we be looking? You're still on mute, Claudia, sorry.
So thank you, Mariam, for the question. I didn't realize they were writing and talking and discussing. So I'm glad. So what's next, and where exactly this parasite, where exactly did it came from? I think this is a question that needs to be resolved. I know the Modric group in Czech Republic is trying to investigate on the origin of the Mediterranean invasions. We are collaborating also with them. It's an open question. It's likely that it came from other regions of Spain, but it's unlikely that we are able to solve this question so far. Where else in Europe it may be present is also something that we don't know for sure. We need to investigate. There is need for more surveillance support. I mean, surveillance is not exactly something that you would always receive funding for. So I think it's important to establish a network for the surveillance of this parasite in the continent, because I mean, we are the demonstration that the parasite can establish very well in this weather. There was a report in Paris in 2016, as far as I remember. They found one human case. There was a lady. However, they concluded that the infection was the result of the importation, the ingestion of imported food. But I have a huge question mark in my mind in relation to this. So I wonder if it's true that it was the reason, or maybe the parasite has been circulating in other regions of the continent. So this is something that needs to be resolved. I suppose other Mediterranean regions nearby us, because they have the same ecology, they have the same species, they have the same weather. They may be susceptible as well. But as I said, more surveillance is needed.
Yeah, that's a very good point. Derek Robinson was actually adding to his question by saying many boats visit Europe from Asia, so it could be recent or old, but remains undetected, and totally linking into your comment about building these surveillance networks. In terms of the patients, there was a question here about whether there were any diagnostic kits available, and there's been actually quite a few answers from Pilar Ferronda from the Canary Islands, as well as Professor Teixeira, Carlos Teixeira from Brazil. So for those of you who would like to find out more, if you just head over and click onto the chat, Professor Teixeira has very kindly actually listed a whole list of resources there. So that's been very useful, having these diagnostic kits to track infection in humans. I'd like to pick up on something, Claudia, that you mentioned, this one word, financing. So obviously there's a huge gaps, huge question marks there. As you say, a lot more needs to be investigated and partnerships need to be built up. And so how have you found the interest of funders in this space? How has your research progressed? And will this kind of importance of building surveillance, would that perhaps elicit and encourage more funding into this space?
Well, definitely. Well, we've been applying for funding. It's true that at the beginning we weren't very lucky, because we only found, at the very beginning, it was only 3 hedgehogs. So part of the reasons of the funders was that they weren't sure that the parasite was actually present. More recently, we have continued finding animals. I mean, we are finding animals positive to the infection for a third consecutive year. So definitely, it's not easy, I think, in general, to obtain money, particularly from my experience here in Spain. It's even more difficult than in some places of the world. We are trying to find funding. So far, we only have a small project for the snail farms, and we are lucky to count with these people that is working extra hours, like my students, or people who's working actually for free to help us. There are students that are, collaborating with us, and many people that is actually interested in helping us. But we still need definitely support. We are applying to several funding sources. But yes, there is need for more support for this type of studies that may be considered basic, but they are crucial to understand what is happening, not only with this parasite, but with many others.
Absolutely. And as you say, not just this isn't just about this parasite, but kind of this whole area of investigating these much lesser known parasites. So would you consider that neural angiostrongylosis is a neglected tropical disease?
That is a really good question. That's a really good question. I have wondered about it. For example, Robert Cowie mentioned this in his blog in Parasite, that because of the lack of funding and the lack of interest somehow in understanding more about the parasite, it's part of the reason why it can be considered a neglected tropical disease. And I think because in many regions of the world, the parasite has been detected, but not many studies have been conducted, we may also be confronting a situation that is even worse than we thought. In many regions of the world, meningitis, the diagnosis of meningitis stopped like that, while the etiological agent is unknown. So it may be happening in many regions. So I think in that term, we can consider this parasite as this disease as a neglected tropical disease. So, for example, in my home country, in Peru, it is in the neighboring countries. The parasite has been detected in Colombia and Ecuador, and in Brazil. So Peru is surrounded, but no more studies have been conducted. So I'm sure the problem is much worse. And many of these regions are developing countries where people may be more susceptible for the reasons I already explained. So yes, I think it is, but it may not be yet recognized by the World Health Organization, but who knows in the next future.
Let's start by getting the word out there that it exists and it is affecting, in a One Health kind of sense, both wildlife, the farming industry, as you said, patients. And I think what you said kind of answers in part a really interesting question by Giulio Di Leo. The question's a bit long, so I might just show it up on screen. But Giulio was pointing out, judging from Kim et al. 2014, with more than 15 species of snails that are competent hosts for Angiostrongylosis cantonensis and so many potential vertebrate hosts, to some extent, it is surprising that the worm is not more widespread. Rats, on the other hand, are a widespread opportunistic species. Do you have a feeling of what has limited the distribution of the rat lungworm? Has something changed in recent years? We had a suggestion a bit further down by Eric, I think Eric Morgan, perhaps climate, or is it just a question of really lack of surveillance?
I mean, I think, it's difficult to know what's the exact number of places where the parasite is present because of the lack of surveillance, of course. I'm sure also climate is a good explanation for the limit of the parasite. It is interesting in that respect that, for example, there was an article by Young. They did this mathematical modeling on the spread of the rat lungworm in the world. And they, for example, calculated that the parasite was going to be present in Europe and spreading in Europe, but they mentioned 2050, because of the limitations by the weather. I mean, we've demonstrated that this happened before. So even when climate may be a limitation, particularly temperature may be a limitation for the spread, I'm wondering as well what is happening with this invasive haplotype. I mean, are we in front of this a particular type of population that may be more prone to establishing in new regions and maybe more resistant, for example, to these cold weathers? It's early to say. It's early to know, but I think it will motivate more and more research, and I think it's important. It's important. So yeah, I agree. There is a lot of there are some open questions that are so far difficult to answer.
Yeah, absolutely. Thank you very much, Claudia. We're kind of coming up to the end of our 60 minutes. There were a couple, well, there were quite a few questions that were left outstanding. I don't know if you have a minute or maybe 5 more minutes to take 1 or 2 more of the questions. Oh, Julia here, Julia DeLeo saying, thank you. I missed the first author of the paper you just mentioned on climate and rat lungworm. So if anyone knows, if perhaps they could just post a link or write the author there, that would be very helpful. Thank you. But while we do that, a question here from Professor Majid Fasihi Harandi, moving a little bit to the patients now. Professor Harandi was wondering whether, moving to the patients diagnosed with eosinophilic meningitis, what is the most frequent clinical presentation? Are there any particular age or gender distributions or other epidemiological characteristics that you've noted?
I mean, here in the Balearics, we haven't had a single human case. In the world, there are many reports. There are many reports in children, I suppose, because of the importance of the parasitic disease in children. I think they happen in children and in adults independently. I'm not sure, but I think, in my opinion, the parasite doesn't really establish, particularly in one particular age group. But I may be wrong because I don't work directly with patients and I don't know much about this part of the research. So many people who is in the audience, like Pilar Foronda and other people can answer it. But in general, I think the parasite can infect many age groups. At least, as far as I know, it's not particularly found in one or other. It is true that it may be present in some people that do not develop any clinical manifestations. Like, for example, I think in Tenerife, they have found antibodies for people. So, I think there is a wide variety of symptoms depending on this is very complex, you know. We have to talk about the host susceptibility and possibly the amount of parasites. It's a very complex situation. I don't know if I have forgotten any questions in relation to this.
No, I think that's brilliant, Claudia. And Stephen Bramer was asking about why the rat lungworm doesn't evolve to the lungs of humans. Professor Teixeira just answered here, saying in humans, the worms are not able to evolve from L5 stage to adults, so they stay in the central nervous system and they do not develop into adults in pulmonary arteries. And again, Professor Teixeira offering lots of answers there to some questions. So it's definitely teamwork today to get our heads around this complicated One Health puzzle. Just to kind of wrap things up, and with a very last question, we talked about funding a little bit earlier on, and you mentioned, of course, the huge importance of the tourism industry. And so there was a question by Major Dillon here, which perhaps would lead us to talk a bit more about whether we can approach this from a different angle, bringing in different kinds of stakeholders or different kinds of funders. But Major Dillon was asking, is there an economic figure about how much damage rat lungworm costs, or is there any kind of work that could try and put this a little bit in perspective and maybe raise the right kind of interest?
Oh, that's a really good question. First of all, I would like to say that I am very honored on the names that you are mentioning. I mean, they are huge names in the field of parasitology and particularly in the rat lungworm. So thank you for attending. And in terms of publications in relation to this, I haven't found a study that would calculate the damaging effects of the rat lungworm. There is one article from Jamaica where they mention the parasite as part of the list of organisms, of parasites and pathogens in general that may negatively affect the tourism industry. I mean, there have been cases in tourists that have traveled to this island. But I haven't came across with any study that properly analyzed the situation. It would be interesting. It would be definitely very interesting. And the touristic industry here is huge, not only in terms of the economy, but there are also some scientists that are focused on the tourism industry. So yeah, it would need it would be an important aspect to analyze. We have actually applied for some funding, trying to answer as well this type of questions. I think it's important to organize meetings with different sectors of the society, but so far we haven't been able to do that yet. Hopefully in the next future.
Brilliant. Well, we're very honored that you also gave us your time to really introduce us. Well, not everyone needed an introduction. There were, as you say, some extremely well-rounded parasitologists in the audience, but for everyone else, we really truly appreciated what for many possibly was an introduction to this parasite, to the ongoing work, the many gaps remaining. So actually, it's been our huge honor, Claudia. I took the liberty, you'd posted your email address at the end of your presentation, so I've taken the liberty to post that as well in the chat. I'm really sorry we couldn't get round to all the questions and all the discussions that have been raised. As Professor Teixeira said earlier on in the chat, there will, for all those interested, there will be an international symposium on rat lungworm disease, which I believe you've posted as being next year. So for anyone who wants to follow up, and of course we will most definitely keep in touch, Claudia, and we will also keep a very close eye on all this hugely exciting and important work. Many of our attendees are thanking you so much. Joan Kirch, very interesting, and thanks for a good presentation. Giulio Delia had to leave. It was an awesome seminar. Thank you all. And Majid, Professor Harandi, thank you very much, Claudia, for your really informative talk. So all around, very big thank yous. Thank you, James, for a nice comment there. And Major Dillon, again, thank you so much. Very informative. It surely is an NTD. We couldn't agree more. So Claudia, many thanks to you. And I hope we can have you back on the ISNTD Connect series really soon. It's been a pleasure to meet you and a pleasure to hear all about your work.
I would be more than happy to do it. And thank you, everyone.
That would be wonderful. Thank you again, Claudia. All the best.